This will be our final podcast on nerves and the nervous system. We'll talk a little bit first about impulse conduction. And you can almost think about impulse conduction along an axon almost like a flame that's traveling down the fuse of a firecracker. And you know from physiology that this is an electrochemical process. So we talk about an action potential with a wave of membrane depolarization. And that depolarization is going to start at the axon hillock. There are voltage-gated sodium and potassium channels here. And as you will learn in physiology, the membrane potential goes from minus 70 millivolts to a plus 30 millivolt potential. The myelination allows for rapid conduction of the membrane depolarization, and this membrane depolarization jumps from node to node in a saltatory or discontinuous manner because at the internodal regions the myelin is insulating the axon, so you don't get depolarization, but you do get depolarization at the relatively bare sites on the axon. So the membrane voltage can only reverse itself where there's relatively little myelin. That's what allows for the rapid and saltatory conduction of nerve impulses down a myelinated nerve axon. Unmyelinated axons, of course, the nerve conduction is much slower. Sodium and potassium channels are relatively evenly distributed along the axon, so the membrane depolarization is occurring in a continuous manner down the axon, and because of that, the nerve impulse actually travels slower. And again, you'll talk about this in much greater detail in physiology. There is a little bit of confusion about terminology with respect to peripheral nerves. First of all, we'll talk about a peripheral nerve as a bundle of nerve fibers that can be held together by connective tissues, and we'll talk about that in just a minute or two. But nerve fibers carry sensory and motor information between the organs and tissues of the body to the brain and to the spinal cord. So exactly what is a nerve fiber? Well, it can have multiple meanings. It depends on how it's used and how it's talked about. We can talk about a nerve fiber as an axon plus the myelin and the Schwann cell. We can talk about a nerve fiber as the axon alone, or we can talk about a nerve fiber as any process of a nerve cell, either axon or dendrite. So multiple meanings to nerve fiber, and of course the peripheral nerve would be bundles of nerve fibers that, as we suggested, are held together by connective tissue. The location of peripheral nerve cell bodies, as we've suggested already, can be within the central nervous system if you're talking about a somatic motor neuron, for example, or if you're talking about part of the autonomic nervous system, the first chain in the synapse. The cell body would be in the central nervous system. The cell bodies can also be within ganglion. So, for example, you've talked about dorsal root ganglion, cranial nerve ganglion, sensory neurons of the autonomic nervous system, for example, might be in those ganglion. You've talked in anatomy about paravertebral and prevertebral and terminal ganglion. Those may contain motor neurons of the autonomic nervous system as well. The image on the left, the two images on the left, show dorsal root ganglion from the spinal cord, both at a low magnification and at a higher magnification. These are, of course, pseudo-unipolar neurons. One way to get a sense of that is to look at the satellite cells that almost completely surround the cell body. Look at the large cell body. You can see the nucleus and nucleolus. Here are some peripheral nerves like this, and look at these little, I just noticed that on this image. Look at the Schmidt-Lantermann clefts like this, the little arrowheads like that. Here itself is the axon. The images on the right would show motor nerves in a ganglion of the sympathetic autonomic nervous system, both at low power and at higher power. Again, a fairly large cell body. Nucleus like this, the nucleolus, and look at, you can see some processes coming in. So these are clearly multipolar neurons. This is just a schematic overview of some sensory and motor neurons. I put this on just as an overview, so this part of the diagram just helps you orient. So just showing the spinal cord with some cell bodies within the spinal cord, within the central nervous system, and then some cell bodies within the ganglia. Here would be some of the axons of these nerves. So here would be the diagram of the nerve. What I'd like to do is talk about some connective tissue components of the nerve, the epineurium, perineurium, and endoneurium. These are connective tissues, so we can talk about an endoneurium. An endoneurium is a loose connective tissue that would surround individual nerve fibers. Allow me to go back for 1 minute. So these would be individual nerve fibers like this. So the yellow around those would highlight endoneurium. Endoneurium is a loose connective tissue. It's composed primarily of collagen fibrils that are type 3 collagen. This type 3 collagen is secreted primarily by Schwann cells. There may be occasional fibroblasts, mast cells, and macrophages within the endoneurium. We can also talk about a perineurium. The perineurium on the diagram is highlighted as this thicker red line, like that. That would be perineurium. The perineurium is a specialized connective tissue that surrounds a nerve fascicle. Bear with me again, I'm going to go back. The nerve fascicle would be collections or bundles of nerve fibers. So these are individual axons, nerve fibers, making up a nerve fascicle, a collection or bundle of nerve fibers surrounded by the perineurium. The perineurium is a specialized connective tissue. It's a metabolically active diffusion barrier to maintain the ionic milieu of the ensheath nerve fibers. It contributes to something we call a blood-nerve barrier in the peripheral nervous system. The perineurium may be composed of multiple cell layers. The perineurial cells have characteristics of both fibroblasts and smooth muscle cells. This is an electron micrograph of peripheral nerve with its perineurium. You can note the single myelinated axon shown here. You can note these unmyelinated axons that would be surrounded by Schwann cells. So this is part of the cytoplasm of the Schwann cells that is surrounding the unmyelinated axons. Here is a blood vessel. There's a basal lamina that's evident on the endothelial cells of the blood vessel and also around the perineurial cells. These would be the perineurial cells that would be surrounding this entire collection of axons. So the perineurial cells here are surrounding the axons, the Schwann cells, and even the little blood vessel. This is a high magnification showing multiple layers of perineurial cells like this. The basal lamina are evident on both surfaces of the perineurial cells. You can see basal lamina like that, basal lamina like that. The cells of the perineurium have abundant rough ER, they have abundant actin microfilaments, and they have pinocytotic vesicles associated with them. You can see tight junctions in the perineurial cells like this, just as you can see tight junctions. This region here is a blow up of this region, just as you can see the tight junctions between regions of the endothelial cells of this blood vessel. So the connective tissue components, besides the endoneurium and perineurium, you have an epineurium. Let me go back a couple of slides. Here's the epineurium like this. Denser connective tissue like this surrounding the entire constellation of nerve fascicles. So let me go back. The epineurium is a dense, irregular connective tissue. It surrounds and binds the nerve fascicles into a common bundle. So if you had an anatomically identified nerve that you would, say, pick up with your forceps during a dissection, the connective tissue surrounding that anatomically named nerve would be the epineurium. The epineurium is often associated with adipose tissue, and it will often blend in with the tunica adventitia of blood vessels. Remember that in your anatomy dissections, oftentimes nerves and blood vessels ran together. So in this cartoon that we just looked at, trying to relate that histologically, here's histological image, and then I've put a diagram down here to help explain this image for you. But on the higher power view here, this would be epineurium that would be surrounding these bundles of axons. Perineurium like this. Perineurium like this. Here are individual axons. A little bit hard to define the endoneurium around the axons, but it would be in a region like that. These might be profiles of Schwann cells. Here's an itty-bitty capillary. Here's a nerve fiber in longitudinal section, a higher power view of the nerve fiber in longitudinal section. Here would be an axon that I'm tracing like that, and you can relate that to the diagram on the left. Here would be the myelin sheaths, so the node of Ranvier, perhaps a Schwann cell nucleus like so. This structure here called neurolemma. Neurolemma is just another name for the myelin sheath that's surrounding the neuron. And here's an interesting histological view. Here's a cross-section of a nerve fiber, and remember in anatomy the nerve itself usually runs in parallel with a blood vessel. Here's a fairly large artery. Here's the nerve. Here's the epineurium surrounding the nerve. Notice all the adipose here, and notice that this connective tissue of the epineurium is blending in to the tunica adventitia of this blood vessel. We can also talk about some afferent sensory fibers, and we're going to expect you to be able to identify these histologically. Two significant ones are the Meissner's corpuscles and the Pacinian corpuscles. Meissner's corpuscles are receptors that are encapsulated with connective tissue. They're usually found within the dermal papillae of thick skin. You sometimes find them on the eyelids and on the lips. These function for fine touch reception. On the other hand, the Pacinian corpuscle is a receptor that's encapsulated with cells. These cells are actually fluid-filled. You find this between the junction of the dermis and hypodermis. In this image, you can see all the adipose, so this would signify the hypodermis. The Pacinian corpuscles sense pressure and vibration. And as we said, we're going to expect you to be able to recognize these sensory receptors in histological images. These receptors, the Meissner's corpuscle and the Pacinian corpuscle, are considered exteroreceptors in that they respond to stimuli from the external environment. There are receptors that are called enteroceptors. These respond to stimuli from within the body. So, for example, you know you've got a full bladder. There are receptors that are responding to the stretch of the epithelium and smooth muscle in the bladder as it's full. And there are also proprioceptors, which provide sensation to monitor the position of the body and muscle tone within space. What are we going to expect you to be able to identify with respect to organization of the central nervous system? We clearly want you to identify regions of the brain and the spinal cord. And the brain and spinal cord, as you know, are protected by bone and by connective tissue membranes called the meninges. The brain and spinal cord, if you want, float in a fluid environment, the cerebrospinal fluid. We want you to be able to identify histologically the cerebellum and the cerebrum. We won't ask you to identify histologically the brain stem, but we want you to identify and distinguish cerebellum, cerebrum, and spinal cord. We want you to be able to identify white matter. And by the way, white matter is given the name white matter because in fresh tissue the axons appear white. So white matter. The white matter contains axons of nerve cells as well as glial cells. You'll see nuclei within the white matter. Those are going to be nuclei of the glial cells primarily. And of course, there are going to be blood vessels in the white matter. So you're going to see nuclei of cells associated with blood vessels. And then the gray matter is going to have a distinct appearance within different regions of the central nervous system. The gray matter are going to have the cell bodies of neurons. Then you may catch the nuclei of the individual cell bodies of the neurons. They're of course going to have nuclei of all the other glial cells. And we'll talk about something called neuropile. That's just the meshwork of the axons, dendrites, and glial processes within the gray matter of those cells. And here's again something to be a little bit careful, because we talk about clusters of cells, clusters of neurons in the gray matter. Those clusters of neurons are given the term nuclei for clusters of cells, but you're also going to be able to see the nuclei of individual cells, individual neurons within the gray matter. So nuclei in the gray matter have two different definitions. This schematically shows how neurons are arranged in the gray matter of the cerebrum. I'm not going to ask you for this course to be able to identify the different layers in the cerebrum, but we are going to be able to expect you to recognize that this region is the cerebrum. I'll show you a histological image in just a minute, and we would like you to be able to pick out these large pyramidal cells. You should be able to easily recognize the cerebrum from the cerebellum. The gray matter in the cerebellum has a very distinct and unique arrangement that's different from the gray matter in the cerebrum. We'll review this in the laboratory, but we talk about the cerebellar cortex as having a molecular layer, a Purkinje layer, and a granular layer. We want you to be able to pick out these Purkinje cells. Here they're shown with a specialized fluorescent stain. They're very obvious and easy to pick out. In the cerebellum, we'll show you a couple of histological images on the next slide of these, but we'll also go over these in more detail in the laboratory. So here is the cerebrum showing different layers. I'm again not going to ask you to identify the different layers, but I think you can pick out the large pyramidal cells in some of these layers. And here is the cerebellum, a very easy region of the brain to identify because of the unique organization of the white matter like so and the gray matter like so. And we'll talk about these layers of the gray matter, but what you look for is the layer of Purkinje cells in the gray matter, which would be found here, which are between the molecular layer and the granular layer of the gray matter. And again, I'll talk about this in more detail in the laboratory. It's a typical profile of the spinal cord, white matter like this and gray matter like this. Allow me to go back for a minute on the brain and look at the location of the white matter is in the deep regions of the brain, the gray matter on the periphery. Same in the cerebrum, the gray matter on the periphery, the white matter deep. Notice in the spinal cord, it's just the reverse. The gray matter is deeper with the dorsal and ventral horns. The white matter is more to the periphery. In the laboratory, we'll go over the components of the spinal cord. We'll talk about the central canal. We'll talk about the gray commissure. We talk about the dorsal horns, the ventral horns. The ventral motor neurons are here. These are very large neurons, very easy to pick them out on histological images. You can see some of the dorsal root ganglion here. This is just a silver-stained region of some of the ventral motor neurons. Finally, I want to take a minute or two and talk about the response of neurons to injury. It turns out that if a neuron is injured, the axon is going to degrade downstream from the site of the injury. Now, in the peripheral nervous system, injury induces axonal degeneration and then neural regeneration. And this is a kind of a step-by-step process, but essentially the axon swells, and as a result, the blood-nerve barrier along the entire axon length breaks down. This is very critical. The blood-nerve barrier along the entire length of an axon breaks down if a peripheral nerve is injured. As the peripheral nerve is injured, the axon swells, the cytoskeleton breaks down, the axon fragments, the Schwann cells will de-differentiate as they lose their axonal contact. As the Schwann cells de-differentiate, the myelin will be broken down, and that will stimulate macrophages to clear it out. And of course, because the blood-nerve barrier breaks down, more monocytes and macrophages can get into the area of injury and phagocytize out the degrading myelin. What will then happen is axon sprouts will begin to reform, and they'll try to grow endoneurial tubes, and these endoneurial tubes will form a growth cone as many of these axon sprouts come together. What will then happen is those Schwann cells will re-differentiate and wrap myelin sheaths around these axon sprouts. That's the actual endoneurial tube. If a sprout and endoneurial tube grows to an effector cell, say for example to a motor neuron going to a skeletal muscle, function will be reestablished. Now, the situation is different in the central nervous system because injury in the central nervous system does not repair because the blood-brain barrier only breaks down at the site of axonal injury. It does not break down along the entire axon length. As a result, the myelin which begins to degrade can't be removed. That's because microglial cells are limited in numbers and they're limited in their phagocytic activity because the blood-brain barrier hasn't broken down along the entire length. Additional monocytes and macrophages cannot get in to degrade the excess myelin. You'll get what's formed an astrocyte-derived glial scar, and the oligodendrocytes will begin to undergo apoptosis, so you can't really repair the injury to axons in the central nervous system. And this cartoon just highlights that. On the right is just showing the limited breakdown of the blood-brain barrier in a central nervous system injury. So the microglia, which are in the central nervous system, are going to begin to phagocytize the excess myelin. Because of this limited breakdown, you get limited amounts of macrophages coming in. So in a sense, a lot of the excess myelin remains as the oligodendrocytes undergo apoptosis instead of dedifferentiation. And you form this glial scar. You don't get axon regeneration. On the other hand, in the peripheral nervous system, axonal injury here, but the blood-nerve barrier breaks down along the entire axon. So look at these monocytes being able to come in, differentiate into macrophages. You get macrophages coming in along the entire length. It's very easy to phagocytize the excess myelin. That phagocytized excess myelin will stimulate dedifferentiation of the Schwann cells as little axon sprouts then reform again. The Schwann cells will myelinate them, forming these endoneurial tubes, and that will allow a tract for a new axon to grow.